We'll continue the addressing, sir. Address screen share, sir. Lateral.
Yes, now. Is it visible now? Hello? ওটা দেখতে পাচ্ছি? Is it visible now? Sound কিন্তু কিছু যাচ্ছে না।
Yes, sir. Is it visible now, sir?
It's visible now?
Yes, sir.
Okay, okay. Am I audible?
Yes, sir. You are audible, sir.
Okay, but here it is not coming that much sound. That's why I'm a little bit puzzled. Okay, please continue, please. Now, we're at introduction.
Good afternoon, one and all. My name is Ramaram from Krishna College of Pharmacy, Bhimavaram. I would like to Professor Tapan Kumar Maiti, Professor and Head of the Department, Jadavpur University, Calcutta, India. Sir has completed his Bachelor of Pharmacy, Master of Pharmacy, Doctor of Philosophy from Jadavpur University. Sir has 30 years teaching experience in various reputed institutions. Sir has 100 plus research publications in various national and international journals. He guided 17 PhD students and currently guiding 4 PhD students. Sir has visiting team member of NBA, AICTE, PCI, UGC, NAC, life member of APTA, member of Society of Ethnobotany, member of Indian Science Congress Association, life member of Indian Pharmaceutical Association, member of BOS for Silchar Central University. Since 30 years, Sir has been working on plants and heterocyclic compounds with their antimicrobial, antioxidant, anti-diabetic, and anti-cancer activity. By virtue of his work synthesis, Sir have completed 2 UGC major research projects, 1 AICTE RPS project, 1 CSIR funded project, 1 UGC UP phase 2 project, purely on drug discovery on plant derived products. We are very fortunate, sir, to have an eminent speaker and with vast experience. All the participants will have great learning experience to this session, sir. Over to you, sir.
Okay, thank you. Hey, ভিডিওটা দাও। ভিডিওটা খুলে দাও, ভিডিওটা একটু ওপেন করো। ভিডিও দেখা যাবে না?
হ্যাঁ স্যার, আপনার ভিডিও দেখতে পাওয়া যাচ্ছে। আপনাকে দেখতে পাওয়া যাচ্ছে।
দেখতে পাচ্ছি? হ্যাঁ। Okay, so good afternoon, everybody. First of all, I convey my regards to Dr. Nataraj, who has invited me. Actually, last 6 years, I have been delivering almost 20 universities, not only in refresher course or some seminar. Last 6 years, I have been delivering your faculty development program as well as academic staff college, as you know. And last 1.5 year also, I have been delivering some NEP programs also. So this is a opportunity to share some views, some news, some of my personal opinion regarding the different ifs and whats of drug discovery and development. As we are in the same fraternity, am I audible? Hello, participants, am I audible?
Yes, sir, you are audible, sir.
Okay, okay. And the slides are visible?
Yes,
Yes, sir, visible, sir.
Okay, okay, fine. So sometimes we shall be discussing our personal shortcomings also. Actually, last 34 years, whatever we have gathered a fraction of something called knowledge or expertise or experiences, we shall be uh, sharing with those with our same fraternity. Either you are a student, or you are a faculty member, or you are from industry, or you are from research or R&D, or you are in analytical laboratories or teaching, etc., etc. As we know that science has developed like anything. If you introspect last 40 years, I think 85 to 90 onwards, the maximum pragmatic momentum has come. So for the uh, advancement of science and technology, and obviously our drug development and drug discovery scenario. Probably you might have already some ideas that how the drug had been discovered in the earlier centuries, particularly in the beginning of the 20th century. There are small discovery or invention was there that we had got a little number of antiseptic, disinfectant, some local anti-infectives like that, right? The father of modern chemotherapy, who is considered, is none other than Sir Paul Ehrlich. It is the it is the invention of 1906 that Paul Ehrlich, who was born in 1854, started his career as a researcher in your 1870 mid 70s, and his hobby or passion, you might be there that the research, innovation, and even the teaching, it does not come in a very lucid manner if you do not have the passion. So passion for research, passion for teaching, and passion for invention. So this needs a huge or lots of pain, struggle, dedications, and perseverance. So Paul Ehrlich, sir, he started to synthesize. You might be knowing that last century, from the very beginning, it was the chemistry of inorganic compounds. The physical chemistry developed a lot, and most of the metal chemistry also developed. Organic chemistry did not develop that much. So for the first few decades of 20th century. Well, early his hobby was to synthesize the arsenical compounds because that time carbon catenations or characteristics were not that much proliferated. So as a result, they have to opt for inorganic compound, particularly some metals or some elements like that. So his hobby was to synthesize, that is conventional synthesis and observing the activity of the drug. So by getting all these one, maximum failure was coming. You might be knowing the fallacy of drug discovery is that failure is but obvious. So in 99% cases, most of them they are failure. Otherwise, 4,500 sulfonamides could not come. 1,000 penicillin could not come, or a report on 84,000 fluoroquinolones could not come. Right? So, Sir Paul Ehrlich in 1906, while synthesizing and observing the activity, he was failed in 605 compounds. 605 compounds did not show any appreciable anti-infective activity. And incidentally, in 1906, 606th compound that was showing some anti-syphilitic activity, a sexually transmitted disease you might be knowing. So that was the accepted drug throughout the world, that STD can be cured with some arsenic compound synthesized by Paul Ehrlich, and this is called arciphenamine, or in our medicinal chemistry it is called salvarsan. So gradually small invention heptagen started, and incidentally all of us we know in 1929 the accidental discovery of penicillins by Sir Alexander Fleming. Right? So, like our poetry says, that little drops of water and little grains of sand make the mighty ocean and the pleasant land. Small discovery may not have that much of impact on the society, on the human life, but whenever all the discoveries come together or club together, that becomes the life-saving drugs. Initially, Sir Alexander Fleming, though he also was failed, and 1939-40, again the research on the invention of penicillin started with the Second World War, and ultimately 1944-45, the first antibiotic in the history of medicinal chemistry, pharmacology in medical world came, that was benzyl penicillin. Though it is a natural penicillin, later all scientists has manipulated the structure, the drawbacks of natural penicillin, and they have come up with the success of semi-synthetic penicillins. And the gift of semi-synthetic penicillins, all of you know, that ampicillin, amoxicillin, cloxacillin, flucloxacillin, dicloxacillin, carbonicillin, nafcillin, right? So these were the invention. Now, invention never be stopped. When penicillin was obtained from one mold, naturally another one mold has been tried for Cephalosporium acremonium, where from scientist Broju in 1962 started to ferment, to isolate, identify and characterize the cephalosporin C, the first naturally occurring cephalosporins. Cephalosporin C, 6-amino alpha adipoyl cephalosporanic acid, right? So in that way, number of cephalosporins came. By the meantime, in the Second World War when stopped in 1945, 1946 streptomycin came, 1947 tetracycline came, 1948 chloramphenicol came, 1959 griseofulvin came, right? So that was the era of antibiotics, because earlier days people did not have a arm to fight the infectious disease. That is why those who were surviving fighting with the disease only due to their immunity or their structural development, nothing others they have to get. Even the doctors were also helpless because they do not have arms or medicines in their hand to treat the patient. Now, 6-amino penicillinic acid, that is a your lead compound. 7-amino cephalosporanic acid, that is a lead compound, as we told. Para-amino benzene sulfonamide, which was the source from Prantosil rubra, by another one, world famous scientist come philosopher Gerhard Domet, who studied and synthesized para-aminobenzene sulfonamide as a sulfur drug, right? In 1945 only, 4,500 sulfonamides had been synthesized throughout the world, based on the para-amino benzene sulfonamide moiety by replacing the nitrogen of N1 and N4, right? So gradually, the development and lead finding, search for the lead finding drugs have been started. Another lead, if I just see, am I audible? Hello,
Yes, sir, you
am I
are,
audible?
yes, sir, you are audible, sir.
Okay. Just if you go back, the drug which has crossed over 125 years, the history goes back. In 1889, a doctor from Germany was treating his ailing father who was having a knee joint pain, and he was giving some extracts with the ointment basis and apply on the knee joints, and further recovered from the pain in general. So immediately that was a plant extract. Incidentally, the plant name was Salix alba. So the scientist, so the doctors, he started to see that what is this your uh, extract there, what is there. Ultimately, it was isolated, identified, and characterized that it is salicin. So salicin is a glycoside. So scientists, they modified the structure of salicin by hydrolysis. They made it salicyl alcohol. By esterification, they made it salicylic acid, and by acetylation, they made in acetyl salicylic acid. So that was the development of 1899. So it was 10 years almost. So the name aspirin or dispirin or acetyl salicylic acid, whatever we are getting till today, not only as a painkiller, but as a cardioprotective drugs also, are drugs platelet aggregation factor and PAPAC and etc. Cardiac patient also nowadays they use aspirin in lower doses. So it is the only drug in the history of medicinal chemistry that which crossed over 125 years. No other drugs are there in the market so far aspirin is concerned. So salicylic acid was the lead compound to get the hundreds of different formulations. You might be knowing the Jandu balm or your pain balm, whatever. Maybe that is having methyl salicylate, alkali sodium salicylate mixture, salol, salicylate, methyl salicylate, sodium salicylate, salicylamide, benorylate, diflunisal. All these are the derivatives of salicylic acid. So salicylic acid became one lead compound, right? Similarly, scientists are curious about the structure. Salicylic acid is nothing but orthohydroxybenzoic acid. In a benzene ring, one hydroxyl group, one carboxyl group. Scientists manipulated the hydroxyl group with their amino group. Its name is anthranylic acid or phenamic acids. Scientists manipulated the structure of phenamic acids or anthranylic acid. They got mefenamic acid, fluphenamic acid, meclofenamic acid, right? So this is a lead compound, anthranylic acid. Just you go to the other natural product, quinine. So quinine is an alkaloid, a quinoline alkaloid, right? When quinine was discovered, you might be knowing Sir Ronald Ross, the life cycle of malarial parasites. He was from Hyderabad, right? Worked in Calcutta also, but later on left for US. In pre-independent period, he asked for a laboratory to, to study the life cycle of malarial parasites. They then British government they did not allow fund for the laboratories, and by sorrows and pain or pathos he left for US, and he did the research work there, and ultimately he is a pioneer man to discover the life cycle of malarial parasites and been awarded Nobel Prize in physiology and medicine, right? So this quinine chemistry, there is a molecule of quinoline. So quinoline has been derivatized, and 1960s onwards, huge number of synthetic anti-malarial drugs came. The mother drug was quinine. The chemical structure of basic pharmacophore was quinoline. Scientists manipulated the structure of quinoline, or so to say 4 amino quinoline or 8 amino quinoline or 9 amino acridine, right? And as it is chloroquine, hydroxychloroquine, amodiaquine, pentaquine, santoquine, isopentaquine, hundreds of anti-malarial synthetic drugs came. So quinoline moiety is a lead compound, right? I think you have lots of examples. If you go on phenanthrene alkaloid, morphine, xanthine alkaloid, codeine, right? Sorry, xanthine alkaloid or purines, or xanthine alkaloid, caffeine, theophylline, theobromine. Somewhere these structures become the lead compound itself. Okay, so in drug discovery and development scenario, there is always a quest how to get a new drug. Now, Sir Alexander Fleming in 1945, when the penicillin was in a hierarchy, everybody was asking for the penicillin to get rid of different disease or infection. Sir Alexander Fleming in 1945 commented, Horace Walpole, one reporter that time, some scientist, he recorded those slogans or those words that the days will come when penicillin will be available like anything in the market, and the people will take the penicillin not sufficient enough to cure the disease, but to produce huge number of resistance. So this is the introspection, outlook, or the idea or the foresight of the genius people, those who can introspect or those who can foresee 10 years, 15 years, 20 years, 50 years later what can be happening. Otherwise, this much of 10,000 formulations are not necessary for curing different infections. But now most of the microbes are resistant to some other antibiotics, not only penicillin. There are so many other antibiotics they are resistant. So this is a problem that human being, when they get freedom, restriction is not there, without understanding or without less without less rationalization, they make abuse of the drug. As a result, your resistance develop. And resistance strain, you might be knowing, they are more dangerous because they itself uh, nullify or neutralize the drug's activity. Rather, the drugs will kill the microbes will kill the drugs, right? So let us see that finding a lead. Now, if you want to have, though some modern techniques tools have come, but still, as you know, that 15 years of rigorous research, nowadays coming to 10 years, I think in near future it may be trying to get 5 6 years. Only the future will say, and the biology of the constitution will say. Constitution means constitution of the human being. They will say whether in 5 years the drug will be coming in the market. So 10 to 15 years of rigorous studies, starting from the chemicals, new chemicals, moiety from isolation or synthesis, right, from natural products or microbes or chemical synthesis, and going to the market as a drug. It may take about 10 to 15 years. Some thousands of crores of rupees involvement. Some 300 or more scientists studying from pharmacologist, biochemist, microbiologist, medicinal chemist, organic chemist, analytical chemist, computational chemist, right? Computational scientist, mathematics, statistics, and doctors overall, and ultimately the patient community. A healthy patient of about healthy volunteers of about 200, ailing patient of about 1,000 for trials. And if they say, I mean the reports say, the feedback say that they are getting cured due to some newer drug, then only the drug will be marketed, provided the safety and efficacy point of view. What is the safety and efficacy? Now, drugs may be excellent in working if the toxicity of the drug is high, the drug will be withdrawn or it will not be marketed at all. Together with paracetamol, there was a drug, phenacetin. It came simultaneously, but phenacetin due to hepatotoxicity has been withdrawn instantly. Paracetamol still there in the market, right? You might be knowing in 2001, rofecoxib and celecoxib came with a high result in the market, very good painkiller, very good painkiller. But you see, after 4 or 5 years, they have gone back almost, right? So there are some problems that safety is the concern, then the efficacy. The drugs will be prevailing in the market if the drug is either active at par with whatever the available drugs in the market, or it is more than that available in the market. If it is less than that, as far the efficacy and toxicity is concerned, efficacy is concerned, they will never see the limelight of the desk of the medicines. Now for a drug discovery, because earlier days, as I told you, Sir Paul Early synthesized 606 chemicals. He expired, he died in 1915. So he's only the invention which came in 1906. So after he only survived 9 years. So just you think of the tolerance, patience, perseverance, and dedication of the people. They have done the research work for the patient. They have done the research work for the betterment of the ailing human being. Right? Now, we are not in that category, or the world will not tolerate that much of poetry. Right? Whole life you'll be going on research, and the outcome will be a problem. Because those who are throwing money for the invention, they are pakka businessmen. Right? They want to get their money, or dollar or franc, whatever may be, to back or to return. So naturally, they will try to get at least one drug in the pipeline. So Pfizer, Ronepole, Novartis, AstraZeneca, right? So these are the German remedies, American remedies. So these are the drug makers, drug inventor, those who produce a pipelines of drugs. Not only your cardiovascular, not only your cancer, not only AIDS, not only your heart disease. And they will try to get at least one or two drugs to be clicking in the market. They will get their money back, right? So now to cut short the research or invention time, the first criteria should be choosing a disease. What kind of disease we want to work. That means we want to work Jew drug will be used by all the houses, all the patients, not only in India, throughout the world, right? Now, if you consider the disease, which are global disease, diabetes is a global disease, cancer is a global disease, heart disease is a global disease, Alzheimer's is a global disease now, right? So these disease, if you go for working a newer drug, obviously you will get exposed. Now, if you go for a third world country's disease, suppose diarrhea, European country you go, less number of cases will be there. USA will go, less number of cases will be there. So maximum in Pakistan, Bangladesh, Sri Lanka, and India, Myanmar. Particularly these SAARC countries will be getting much more little bit, except Japan also will not be there. Korea also will not be there. Even China also will not be there. So generally 5-6 countries, they will be absorbed with this anti-diarrheal drug, anti-amoebic drug. So if you go for research with this drug, it's very difficult to get a worldwide reputations, because obviously the global demand is high, right? Coming days, the neurology drugs will be capturing the market like anything, because the days are coming, people are full of stress. From the very beginning of the birth, if you consider, people are getting or people are under stress. So that has to be that we have to think of that choosing a disease, what kind of disease we shall be working. So choosing a drug target, where the drug will be targeting, The drug should target either you might be knowing all of us, drugs should target either a protein or a receptor or a DNA or RNA, whatever may be. So if it does not match with the infective organisms, fine, because then selective toxicity will come. If it matches, that is the problem. That is the only fallacy that anti-cancer drugs so far. I know there are huge advancement of anti-cancer drugs, but till today, only targeting the cancer cells, not the normal body cells. If it once it is finalized, cancer will be cured, right? So choosing a drug target is very essential. And you see the antibiotic, how that does they work. Antibiotic, the cell wall synthesis of the bacteria, cell wall synthesis of the human being are different. That is why easily the drugs are targeting the cell wall synthesis of the bacteria, and we are getting selective toxicity. A drug target, if a drug or a poison produces a biological effect, the drug is a poison and poison is a drug, right? Earlier days, mercury, antimony, iron, lead. In the homeopathic medicine, people used to have this one very light concentration of the drug as a drug. But we know that they are very poisonous, right? So drug in higher dose will be a poison. Or poison in a very, very less dose, if you consider, they can be a drug, right? So they must have a target. When snake bite is there, snake venom is there. The venom finds some sites in the human body. That means some receptors are there which can be coupled with the venoms. That sites are required, right? Morphine was accidental choice because the morphine receptors, so to say, opioid receptors are there in the body. It's because since long ways, hundreds and hundreds of years back, people used to have opium, which is a food also. We know, posta, papaver somniferum, the seeds. It's a food, spices, all of you know. But again, from there, the extract is offing, right? And morphine is there, the phenanthrene alkaloid, which is a narcotic analgesics. Now, if you make it overdose of morphine, people will die. Very less dose of morphine is a unique and excellent narcotic analgesics. And based on this morphine molecule only, hundreds of non-narcotic analgesics as well as some antagonists also have been designed. Right? So drug target is very essential, that in which part of the body the drug should focus. Now target specificity and selectivity between species, that I told you that penicillin targets the human bacterial enzyme or bacterial system, cell wall, not the human being. So if the specificity is different, sulfonamides, right, they interfere with the PABA, para-amino benzoic acid synthesis. In due course, that PABA to your folic acid, to folinic acid, dihydrofolic acid, to tetrahydrofolic acid, then cell. And the sulfonamide blocks the PABA because they are the structural anomers, look like same similarity to the para-amino benzoic sulfonamide. So sulfonamide is acting, antibiotic is acting, right? So target specificity and selectivity between species is very, very essential. Within the body, see, our system is in such a manner because we are all the natural products, right? Now, somewhere you see both the hormone, acetylcholine, adrenaline, dopamine, GABA, right? All these are side by side, insulin, glucagon, right? Cortisone, hydrocortisone, if it is there, there are so many others also, right? So enzymes, proteins, hormones, they are may having some different activity, but very dynamic equilibrium. They are in our body, in our brain, in our organs. And when the situation arises, some supersede others. Then only the problem started. Otherwise, they are in a dynamic equilibrium. As all of us know, insulin, glucagon, they stay in the same cells. I mean, same organs, but they are in a very conducive environment they stay. But whenever a diabetic condition comes, generally insulin activity started and glucagon activity will be there. That is their interference. Adrenaline, noradrenaline, acetylcholine, adrenaline, right? So these type of things, right? So variety of receptors, histamine, antihistamine, agonist, antagonist, adrenergic, anti-adrenergic, cholinergic, anticholinergic, cholinesterase, anticholinesterase, right? So this is a uh, our system may have that kind of things, that in the necessity only something will supersede the others. Otherwise, they are in a very dynamic equilibrium condition. So whenever a drugs act, they should not selectively act for a particular receptor, but also to the subtype of receptor. H1 receptor, histamine, H1, H2, H3, like that, right? Kappa, mu, delta, gamma, this type of receptors. They have to have pitfalls. What is the pitfall? Now, our body, if you say, our body is not a pipeline, right? That you put the water, 1 liter water this side, that means another 1 liter water will be going outside, right, in the pipe. If you take 1 liter water in the body, it will be utilized, absorbed, all these things, and ultimately in excretion from 1 liter, maybe 200 or 300 ml. Others will be constitutions of making the body's equilibrium, electrolyte balance, and everything, right? So body is not a, pipelines. It's a complex system. Otherwise, if you see that there are so many messengers, receptors, enzymes are involved. Suppose if say somebody drug targeting, because now all of us know drug targeting is the slogan of nowadays, particularly bedside medicine, patient-oriented medicine. Uh, there are, I think, those who are PharmD, they will be much more rigorously going for this training. That in a family, maybe 4 people are having 4 different drugs treatment scenario, or maybe for same disease, right? So targeting, we are telling that targeting cancer, targeting diabetes, targeting cardiovascular problem. So targeting cardiovascular problem, maybe one drug could be sufficient to arrest the cardiovascular abnormalities or whatever may be the problems. But have you seen that any cardiovascular patient is taking only one medicine? You'll say multiple cocktails, because drug targeting is not like that. Just you put it and it will be just directly targeting the particular cells of target. It will be targeting the cells simultaneously. The surrounding cells also will be hampered, and as a result, some side effects may be there. Right? To address the side effects or some toxic effects, you have to put another one neutralizing substance or drug. Right? So as a result, a number of drug target has to be addressed instead of only addressing one target, because our body is a complex network. Right? We have some thousands and thousands of neurons. Okay, you just open the, in your house, if you open the TV, the back of the TV, circuits, right? I see. You open your mobile in front of somebody, open the mobile, hundreds of circuits, right? Then only mobile functions. In our body, thousands and thousands like circuits, neuronal networks are working. And as a result, we are laughing, we are walking, we are laughing, we are talking, we are working. And one day or other, if these neurological networks are jeopardized, then we are dead, right? So body is a different manner, right? It's not like that. That's why that is a pitfall. If only the drug target cancer drug, means only cancer cells will be destroyed. There is no harm of the normal body cells. If it is there, it could be, obviously one day or other it will come, because science is developing and going a lot. Then cancer will be cured, right? Not controlled or treated or managed like diabetes, right? In vitro tests do not involve animals, instead specific tissues, because we know animal working on the animals, it's a very hard task and you have to have lots of protocols to be followed. Cells or enzymes are used. Enzyme inhibition can be tested on the pure enzyme in solution. The affinity of drug receptors, how strongly they bind, can be measured by radioligand studies, radioimmunoassay. Radioligand studies, how the drugs are binding particularly, right? So in vitro test, because we are not sacrificing the animals, only the cell lines or the enzymes. In vitro test, it becomes easier nowadays. Earlier days, only directly you are administering in the animal, maybe your uh, mice, rats, or rabbit, whatever may be. Then we are observing the changes in the animals. That is why in the pharmacology, either Goodman-Gilman or Lewis or Cranston and Carr, right? If you just see that they have action on CVS, action on CNS, action on urinary tract, action on like this. But it is not a, it is a generalized. It's not a particular, right? But in vitro assays, nowadays it has given huge tests. A particular cell line or enzymes you put, and that's why high throughput screening has come, HTS. In vivo test, rather, this will be authenticated next stage of development, because from in vivo only you will go from animal testing. Sorry, human being trial. Animals often involved in using clinical conditions to produce observable signal. We know that anti-inflammatory activity, analgesic activity, right? Potentiation of the barbiturates, sleeping time, sleeping your swimming endurance test. See the activity, right? That means we put some chemicals. It's very difficult to get a disease animal. You have to induce the animal with particular disease or disorders. We put streptozotocin for diabetes or alloxan for diabetes, right? And different substances are there for intoxications. And then we see the observable symptom, and that observable symptom, how to alleviate with our drugs. Then we check it in vivo test. Test validity, antibacterial agents can be tested. We know that culture sensitivity tests are there, right? That will give that which bacteria, which antibiotic is much better than others. As I told you, high throughput screening. Just you think of what Sir Paul Ehrlich has done, 505 compounds, almost 25, 30 years, right? 30 years means one's life is over. So now within few years I will be retired also. So now already 35 years means we have synthesized hardly about 100 compounds, right? That is a 505 compounds, right? So it's a huge time wastage, you can say. I should not say for research it is wastage, but that much of time investment. That is why high throughput screening has come. The microplate reader, the multiples of 98 holes or cubes, plates. So 98 receptors, one drug, 98 drugs, one receptor, or mix up. That means one day you can go for a testing of 98, either it's a new compound or it's a derivative, whatever maybe. Or earlier days for 98 compound, whatever we have say for conventional synthesis, at least some years you have to work, right? So high throughput screening, yes, it has cut short our research timing and wastage of time also. Because earlier it is there was no other avenues, people had done it. Nowadays the avenues are there. We have to go for that. Now screening by NMR. Screening by NMR, simply whether a compound binds to a protein targets can be detected by NMR studies. The NMR spectrum of the drug is taken, then protein is added and the spectrum rerun. If the drug fails to bind the protein, then its NMR spectrum still detected. If the drug binds to the protein, the spectrum will be different, right? So drug protein binding, if it is there, that we have to see. Okay, individual NMR of the drug, individual NMR of the protein, and at the administration of the drug, right? If you see the NMR of the complex, if at all it is there, if the same NMR comes, that means it has not been binded. If the NMR changes, it has been binded. So screening by NMR also can lead some pathway for the drug discovery. Once a target and testing enzymes have been chosen, the next stage is to find a lead compound which shows desired pharmaceutical activity. The lead compounds provide start for the drug design and development process. Yes, target is ready, testing system is ready, right? Then lead compound, because lead compound is a leader compound. A leader can inspire hundreds of people. Similarly, a leading compound or a leader compound, from there we can prepare or we can synthesize a medicinal chemistry, organic chemistry particularly, hundreds of compounds they can synthesize. Screening of natural products, as I told you that Foxglove, digitalis, dioxin, chitoxin, chitallin, right? Argometrine, argocristin, argocryptin, these are natural products. From their screening, number of compounds have come. You have seen probably that digoxin steat, it is a life-saving drug, a cardiotonic glycosides, cardiotonic drugs, right? The secondary metabolites, they have complex structure. Yes. Just you think of the bacteria containing the drugs, particularly antibiotics. If you see the structure of streptomycin, if you see the structure of erythromycin, just you see, can you synthesize it? Is it possible? No. By debate, you can say, sir, it is possible. Hundreds of steps may be there. But as you know, so hundreds of steps mean hundreds of impurities. There is a chance. But the bacteria, the microbes which are not seen visibly, I mean naked eye, they contain such a huge structure. The students, the participants, if you have the mobile, just you can open the structure of erythromycin, roxithromycin, azithromycin, clarithromycin, thromycin, spiramycin, mitramycin. You see the structure, you will see that this structure probably the chemist, a medicinal chemist, cannot dream of synthesizing, but scientists have isolated those molecules and established. Natural product is a huge source of. The plant kingdom have been always a rich source of lead compounds. Many of these are useful drugs in themselves, and others have been basis of synthetic drugs. Paclitaxel, Taxol. You might have known that ovarian cancer, particularly breast cancer, they are being very nicely treated with Taxol. Esescitabine lactone, obtained from Texas brevifolia and Texas baccata, right? Those who are chemistry person or pharmacology person, you can check it from Burgess Medicinal Chemistry or your, what is that, your Goodman Gilman Pharmacology. 38,000 adult Texas brevifolia tree growing for more than 2 years. If you chopped it, take the raw material, you will get 25 kg Taxol. 38,000 trees, just it's not a matter of joke. Chopped it and get 25 kg Taxol. That is why Taxol is more than gold now, right? You can say Taxol is very costly. Yes, it's a very, very good drug. Artemisinin. The 2016 Nobel Prize came for artemisinin. UU2, one scientist got Nobel Prize from Artemisia annua, developed in China, collaboration with US and Japan, and ultimately the physiology and medicine awarded this Nobel Prize to this artemisinin alkaloid, a Plasmodium falciparum, that is cerebral malaria, a drug from cerebral malaria, because thousands and thousands of people have died due to this resistant malarial germs, falciparum, right? And artemisinin was the remedy for that. Artesunate and artemether or artemether, the semi-synthetic derivatives, because it is very unstable compound. So the ethereal compound has been only stabilized by RT ether and RT mether, either methyl ether or ethyl ether. They have been made. This is also a terpenoids compound. Artemisinin, it's an alkaloid, right? The microbial world, what I will say, penicillin, cephalosporin, tetracycline, streptomycin, gentamicin, kanamycin, neomycin, right? If you say cephalosporins, all are from microbial world. Yes, there is so much possibility of the marine world. Probably you might be knowing the teprotide, right? The biostatins we are taking nowadays. Those are cardiac failure or some problems are there. Almost almost in a family after 10 years, 15 years, every people will be taking statin, right? Dolostatin, biostatin, or atorvastatin. It means surovistatin, something like that. This is also kind of statins. Furacin anti-cancer drug, right? Cyanobacterium, other anti-cancer, anti-tumor agents. Eleutherobin, discodermolide, bryostatin. These are all marine world. So marine world as working in the plants, working in the minerals, working in the uh, microbes. To some extent, if you say, it is little easy compared to working on the marine source, because collection is a challenge. Collection is a challenge. Then preservations and getting huge amount. It's not like that the plants are available in your field or in your village. You go and collect it, then identify it and go for extraction, isolation, etc. Going for the seabed or ocean bed, it takes lots of time, right? One minute. এই একটা ক্লাসে আছে, একটু পরে ফোন করো, হ্যাঁ? So the marine world, it is a huge potential. So this world, if you say the 3/4 of the earth crust is land and sorry, 1/4 is land and 3/4 is water. Now underwater 3/4, just you think of huge sources are there. I think in near future more and more uh, life-saving drugs will be coming. Animal source, a series of antibiotic peptides were extracted from the skin of African cloud frog, and a potent analgesic compound called epibetidine was obtained from the skin extracts of Euclidean poison frog. Yes, even in our school level, while college going also, we had seen that the frogs are huge demand. Some agency people are coming to the village. They were asking 50 paisa will be given to you if you give us one frog, right? And as a children, we are very much happy to get 50 paisa and to give some frog, because in the village area particularly, huge amount of frogs are available in the rainy season particularly, right? So sometimes the frogs, even viper venom, right? These are very source of huge anti-venoms, I mean drug, you can say. Venoms and toxins have been used as a lead compound for the development of novel drugs. Teprotides, everybody knows, right? A Brazilian viper, snake, a lead compound for the development. Yes, in the market now you see captopril, enalapril, silajappril, all are there in the market. That's why the cost of snakes, you might be knowing some news are coming, either in the airport or the seaport or the railway stations, right? That huge number of snakes have been found getting exports on other countries or something like that. The customs or excise department, they have caught hold them, and police has arrested, something like that. This is nothing but they are very costlier for the research purposes. Medical folklore, if you consider, because sometimes we very happy becomes that medical folklore, our India, our Bharatvarsha, you can say, we have lots of information. Unfortunately, documentation is not there. Only the Shruti, so to say, by enchanting or by listening or by rhyming or by remembering, it's very difficult. Traditional Chinese medicine, TCM, they have 2000 years old materia medica. I mean, after the Faiyan or Yuan Sun, if you say the Chinese travelers, They have 2000 years old materia medica. And India has such a vast country having vast biodiversity. Even your Telangana states, right? The coastal weight, particularly your Rajmundry, that side, if you what is it, that side, Acharya Nagarjuna University, what is Anantapur, na kya bolte hain? I forgot. So that side is a huge biodiversity is there, right? Unfortunately, our materia medica only written in 1923-24. That too, a doctor called Dr. R.N. Chopra and I.C. Chopra. Particularly R.N. Chopra was posted, he was a doctor, he was posted at Jammu and Kashmir in undivided India, right? And he was there working in that. Later on, that laboratory was converted to RRL Jammu and Kashmir. And later on, 2005 or 2008, I think, that has been converted by, undertaken by CSIR lab in the name of Triple IM, Indian Institute of Integrated Medicines, right? So R.N. Chopra, just how much pain he has taken. He used to mix up the tribal people, the rural people for getting the folklore information and compile the books, that is treatise Indian medicinal plants, treatise on Indian medicinal plants. R.N. Chopra, that is a materia medica, Indian materia medica, right? But everybody knows morphine is having used, caffeine is having used, your Vasoka has been used, Cascara sagrada, Jalap, Colosynth, Polophyllum, right? Senna, Cassia tura, Cassia occidentalis, Cassia tussilifolia. People know these are purgative drugs, right? But it is only known from listening. Their documentation started in maybe from the very beginning of 2000, I mean 1925, 26. That is the issues that we are lagging behind to losing some uh, patents of our old traditional medicines also. You might be knowing there are huge tassels with turmeric, right? The turmeric is not from India, turmeric is from other countries. People has made it because they are having advancement of thinking also. Rhubarb roots contain anthraquinone glycoside, right? Anthraquinone, the three benzene ring containing compound having two ketone groups in the 9 and 10 position, anthracene dione is called anthraquinone, right? From there, sinucide A, sinucide B, anthrone, dianthrone, amodine, alloamodine, hundreds of alternative structures have been there. It has been isolated. They are purgative drugs. Even they are anti-infective as well as anti your skin, in the skin eruptions they can be used. Ancient record of Chinese medicine provided that due to the novel anti-malarial drug artemisinin, as I told you, opium poppy, it was there in the Egypt earlier days also. Atropine was used by the Greece and earlier also. From there, hyoscine and atropine has been isolated. Snake root poison, that is reserpine. The days comes, the research changes, right? Reserpine earlier was a snake repellent. Later on, reserpine became the your what is that, major tranquilizer, or I will say antipsychotic drugs. And reserpine later on came as a anti-hypertensive drugs, right? Willow tree salicin, and Foxlow principle digitalis is a mixture of digoxin, etc. etc. The cardiovascular drug, salicin is analgesics. Screening synthetic compound libraries to know, because nowadays it is very easy. Compound library, particularly the QSR and drug design, where compound library is very, very easily available by the softwares. The crores and crores, thousands and thousands molecules are available, I mean virtual at least. From them you can select calculation or different descriptors by different analysis, and you can select some compound for the synthesis. And companies purchase novel compounds prepared by research groups elsewhere, a useful source of revenue for hard-pressed university department, right? If you see, in foreign countries, so many university departments are sponsored by the industries. And the industries, they are expecting some know-how, because this is a workshop, the university laboratory, because we do so many things, but we do not have the arms or financial support to go for the latest advancement if you do not have that commercial viability by patent, I see. Right? So this awareness has already been there maybe 50 years, 60 years back in European countries, 70 years back. But somehow last 10, 15 years, there is awareness for patent. But patent should not be only for the certificate for patent. I have lots of patent for getting my CV very grave. That's lots of patents are there. Patents without any commercial viability, they does not have any value, you can say. But your work is worth, agreed. But patent without any commercial avenue, if you say, that number of patents, obviously some will be coming in the near future as usable tools. Existing drugs, me too drugs, you have seen that probably that there are so many drugs which have been derivatized, and recently if you see the Pharma Times or our CDSCO guidelines or some notices that some drugs have been manipulated or modified to have the same category of another drug, a new invention, bypassing the your what is that, your patentization. So as a result, because already useful drug in the market means the structure is known, everything, that basic research is done. Already you are getting the market, drug, ready-made drug. Now you modify the drug in some derivatives, and you can bypass the patent restrictions that no, your drug is not my drug is having methyl group extra or ethyl group extra, nitro group extra, amino group extra, so we do not have, right? They can modify, right? So me too drugs, it is there that antihypertensive, the captopril was used as a lead compound by various companies to produce their own antihypertensive agent. So captopril is already there in the market. Now this captopril has taken as a basic molecule. Now you modify. Enhancing the side effects sometimes. Yes, sulfonamide have a side effects, convulsive effect, but by hypoglycemia. So sulfonamide is having a side effect, hypoglycemia, right? Now, in a diabetic patient, hypoglycemia is required. That is why sulfonamides, you might be a tolbutamide, glibenclamide, acetohexamide, glipizide, sulfonamide derivatives, right? So they have been developed from the side effects of sulfonamide to have a good drug for diabetes treatment. Natural ligands for receptors, I think obviously 5-hydroxytryptamine antagonists, sumatriptan, histamine, antihistamines. These are already there. Natural subs for enzyme, that is also there. Modulators as a lead compound sometimes, benzodiazepines, right? Gamma-aminobutyric acids binding to the allosteric binding sites. I think so far benzodiazepines are what not. Sedative hypnotics, anti-convulsant, psychotic drugs, antipsychotic, neuroleptics, psychedelics. There are so many use of this drug, right? Combinatorial synthesis, I think nowadays straightforward, people are trying to make it, but combinatorial, that means from all angles you will go for the synthesis, and if there is a possibility that you can get a number of mixtures, isolate those mixtures and get some drug, and computer-aided drug design, I think this will be reducing the expenditure as well as the unnecessary synthesis of the drugs. At least, though there is also success, there is a challenge, because what computer CAD design, they say this kind of drug you can go for synthesis. Some of them are coming, some of them are not at all coming in synthesis. So virtually available hundreds of molecules may not be possible to have been synthesized in the laboratory, because ultimately the synthesis is the word which can be done. Whatever the artificial intelligence you will be using, right? Human brain will be there. Yes, it can be helping. Robots helping us, machines help us, computer help us, laptop help us. But your emotion, your originality, your habit of reading, remembering the things, that will come from your own. Yes, last 10 years, artificial intelligence, machine learning, all these things are, this is also a machine learning. This is also intelligence, yeah, obviously. Yes, it will reduce the time slot. It will reduce, or it will sometimes it will help to your determine the drawbacks or lacuna where it is. It will be helping, obviously. And we are hoping that, yes, time spent from drug discovery scenario will be your drug development scenario. Solubility is not proper. Why it is not there? Artificial intelligence might be telling us, then how can you make the solubility much more better? As I told you, paclitaxel and artemisinin, there are the solubility issues, right? So computer-aided drug design, it is good one, but still structure of the protein and its binding site by X-ray crystallography only will be categorized and will be taken authenticated. We have published a paper in RSC, medicinal chemistry. We have to give a single crystals particles and their crystallography. Single crystal. How much toiling we have done. Really, synthesis there is also a huge challenge. Serendipity and prepared mind. This is the drug discovery scenario. You may plan so many things that things may not come. Right? Cisplatin, penicillin, serendipity. Clonidine was originally designed to have a nasal vasoconstrictor, running of nose and all these things, and nasal drops. Clinical trials revealed that it caused a marked fall in blood pressure. Now it is an antihypertensive drug. So clonidine started with a nasal drops. People were happy with that, but other result came that it's a good antihypertensive drug. Aminoglutathione was prepared as a potential anti-epileptic drug. I am just giving some example. Now it is used as an anti-cancer drug. Anti-impotence drug sildenafil citrate. You might be knowing in the name of Viagra. Hundreds of males have come, right? The anti-heart drugs therapy, the project in US, they were coming in a huge hierarchy. That sildenafil citrate is a very good drug, right? Anti-impotence to have all the sexual arousal in the human. So by secrecy, people were using in different way. Unfortunately, majority those who have used, some of them died of heart attack itself. So the drug which are supposed to be heart protective, induction of the heart attack itself. So sildenafil citrate is not a marketed drug, but behind the screen it is being marketed in different way by peddling. Isoniazid was originally developed as an anti-tubercular drug, INH. Patient taking it proved remarkably cheerful. Those who are taking laughing gas, you might be knowing also, right? A series of antidepressant drugs known as a MAO inhibitor, right? Laughing gas. We use MAO inhibitor. That those who are suffering from depression, right? For them, antidepressant drug is to make them cheerful. Thank you. So I think it's a little bit much more time I have consumed. So I may be stopped here, right? Natraj sir, hello.
Thank you, sir. Thank you. Thank you, sir.
Hello.
Thank you, sir. Sir.
Sir, audible, sir. Yes, sir.
You are audible, sir.
Okay, I am over. Please.
Yeah, fine, sir. Fine, sir. Thank you, sir. Thank you.
Any questions from the audience?
Okay, fine.
Your participant having any questions, please ask. Okay, sir, I think there are no more questions. Hereby, I'm going to give the vote of thanks, sir. Good evening, all. So my name is B. Brahmaya, working as assistant professor in the Department of Pharmaceutics. And then good evening, Dr. Tapan sir. And it's great honor to propose a vote of thanks on the behalf of the National College of Pharmacy, Bhimavaram, on this 1 week online national FDP 1, that is on challenges and innovative solutions in the drug discovery and development. I extend my heartful gratitude to our speaker, sir, Dr. Tapan sir, for sharing valuable insights with us. Thank you very much, sir. Thank you, Tapan sir. And a big thank to all the participants and all the active engagement and the participation in the FDP. Thank you all. Thank you once again. Thank you all.
Thank you, sir. Thank you, Tapal
Thank
sir.
you.
I'll get back to you, sir. Thank you. Thank you, sir.
Thank you. Okay. Bye.
Very